Good morning, everyone. Welcome to the Research Coordination Network SARS-CoV-2 and Wastewater webinar series for junior researchers. The goal of this webinar series is to give all the hardworking assistant professors, postdocs, and graduate students time to talk about their research and celebrate their hard work during the pandemic. So thank you, everyone, for attending. Today we have two speakers, Sarah Philo, who is a PhD student at University of Washington, and Bo Lee, who's a postdoc at University of Hawaii. And Sarah will be going first. Sarah, you can go ahead and share your screen as I finish the instructions here and announcements. First I want to mention how the Q&A works. Please put your questions into the Q&A tab at the bottom of the webinar. We are not monitoring the chat, so if you put a question into the chat, it won't get read. So please put it in the Q&A, and at the end, towards the end of the webinar of each person, please upvote questions that you'd really like to hear answered, and the moderator will ask those questions first. I also want to mention that we do record all of these webinars, and they are available on the RCN website, which Jill is going to put into the chat. And with that, I think we'll just hand it off to Sarah to begin.
Hi, again, good morning. So as the title of the talk suggested on the website, I was going to go over some of our recovery organism comparison work, but most of that is just getting started. So instead today I will be talking about the SARS-CoV-2 surveillance we've been doing in Seattle area wastewater. So first I will talk about our sampling locations, and then I will go briefly into a methods comparison that we did back in the late spring and early summer. And then I will talk about a method development that sort of grew out of that methods comparison, and then I'll present some trends in our SARS-CoV-2 data. So first, this is a map of King County. We have been conducting weekly sampling since late March of 2020, so we're going on almost a year now. The bright pink dots is the three wastewater treatment plants where we were collecting samples from. We have been conducting skimmed milk flocculation with and without virtual extraction for every week since then, plus a few other methods sprinkled in throughout there. The three treatment plants we've been collecting from are West Point, South Plant, and Brightwater. And this is a map of the water catchment areas for those three treatment plants. West Point has about 100 million gallons per day flow rate, which is the largest in the area, compared to Brightwater, which has about 18 million gallons per day of an average flow rate. The populations that these serve is also pretty different. So West Point served about 769,000 people, South Plant served about 815,000 people, and then Brightwater served about 259,000 people. So for our methods comparison, we conducted a 15-week seeded methods comparison from March to July of 2020 using human coronavirus OC43 as a seeded recovery control. I'll only go over this briefly since it was published, but it did help inform a lot of what we have continued to do since then. So we collected our wastewater from three Seattle area treatment plants, like I mentioned before, and then we seeded OC43 at roughly 3.3 times 10 to the fourth TCID 50 per liter. We picked OC43 because we felt it could be a good representation of SARS-CoV-2 because they're both human respiratory viruses. So we thought that it might be a better representation about how SARS-CoV-2 might behave in wastewater. And then we compared 4 different methods with the seeded organism. So we first compared the bag-mediated filtration system, or BMFS, that Scott Group developed for polio surveillance. And then we did skimmed milk flocculation. And then with both of those methods, we did do virtual extraction on half of the sample. And then we did polyethylene glycol precipitation and ultrafiltration. We also ran a direct sludge extraction, but we did not seed with OC43 for that. And then after concentrating the wastewater, we extracted these concentrates using the QIAGEN viral RNA mini kit, and then ran RT-qPCR using the CDC assays. So this is just some brief descriptive data of how those methods went. So we first looked at effective volume assayed and OC43 recovery. So the effective volume assayed is the percent of the original wastewater volume that was actually assayed by PCR after all of those concentration steps. So both BMFS methods perform pretty well for volume assayed, in part because of the large initial volume used, concentrating down to a really small concentrate. But they didn't perform as well when we looked at the actual recovery of OC43. So we think that this could be because not only are we also concentrating all the viruses, but we're also concentrating all of the inhibitors that would make PCR less efficient. There could also be some problems with the filter chemistry not being the best way to concentrate enveloped viruses. But even though skimmed milk with and without Bertrell have lower effective volume assayed, they have performed pretty well for the recovery of OC43 compared to the other methods that we ran. And then in terms of how these methods did for SARS-CoV-2 detection, so the size of the circles is the number of samples we ran, and then the darkness of the red is the percent positivity for SARS-CoV-2. One thing to note here first is that we didn't have the same number of samples for each method, and there were a lot of reasons for this. First was just personnel limitations on how many people we were allowed to have in the lab. It is a pandemic and there are restrictions on the number of people we can have, so that sort of limited our ability to run a lot of methods every week. We were also limited by machine availability, so most of these methods require centrifugation in a large centrifuge at different speeds and different temperatures, and that made it hard to find time on the centrifuge given the number of people we had. And then we also had some supply chain constraints, which I'm sure a lot of us have had as well. The ultrafiltration centricons were pretty hard to get a hold of and are still pretty hard to get a hold of. So we ran as many methods as we were able to find for the centricons. But we do have the BMFS and then both skimmed milk methods for all 15 weeks. One thing to first point out is sludge seems to perform pretty well for SARS-CoV-2 detection, that's sludge extraction, but running the kit takes a particularly long time. So we did not continue with that after we finished the methods comparison. And then the method with the next highest SARS-CoV-2 detection across all 3 treatment plants is both skimmed milk methods. So for this reason, we have continued to do skimmed milk flocculation weekly since that point. But this set of experiments led us to have a lot of other questions. So primarily, how can we speed this process up? Skimmed milk flocculation does take a whole day and leaves little time to continue on with extraction and PCR. And ultimately, the goal would be able to run concentration all the way through PCR in a single day. And then sort of going along with that, because it takes a whole day to concentrate, we have quite a few freeze-thaw cycles between concentrating the wastewater and actually running it on PCR. So it's likely that we are losing virus each time we do those freeze-thaw cycles. And along with that, we're interested in thinking about how can we increase recovery of the virus. So this sort of led to a little bit of a method development. So we have been doing a direct pellet extraction of those skimmed milk pellets to try and speed the process up. So how this works, we do the normal skimmed milk flocculation where we seed with human coronavirus OC43 and then add a 1% skimmed milk solution or a final concentration of 1%, and then we spin it or we shake it for 2 hours and then centrifuge. But instead of going into a resuspension in PBS at that point, we resuspend those pellets in the lysis buffer from the viral RNA mini kit. And then we transfer those pellets to a power bead tube and bead for 10 minutes. And then it just continues on with the normal protocol for the viral RNA mini kit. There are a lot of benefits to this method. It takes substantially less time to get RNA than the traditional skimmed milk protocol. So it takes about 3 hours of active lab time to get an RNA extract compared to about 6 hours for to get to an RNA extract for the traditional skimmed milk method. It's also more scalable because we only use 100 mils of wastewater instead of 500, so we could run a lot more samples in a single day compared to the skimmed milk protocol. So how does this method compare to skimmed milk? First, it has a much higher volume acid, so it's a little hard to see in that, so I put an arrow here, but it's 6.25 mils every time you run it, and because it's the same volume, you can get a really consistent volume assay for each sample. And we still have fewer replicates at this point, so the recovery isn't exactly comparable, but it looks similar to the other skimmed milk methods, and we're ongoing with this just to get some more data to see how it compares. And then the other thing to note is it actually does pretty well for SARS-CoV-2 detection as well. It has pretty comparable detection for SARS-CoV-2 at both South Plant and Brightwater. And it detects SARS-CoV-2 at much higher rates for the West Point water. So this could be either due to matrix effect. The water is pretty different. I know all of us that have worked with these samples, West Point usually doesn't have as many solids as the other two. So that could be a reason. And there could be differences between the methods for those plants as well. And again, this graph, like before, the darkness of the red is SARS-CoV-2 detection, and then the size of the circle is how many samples we've run. But I know the big question that we're all trying to answer is how our data relate to COVID trends in King County. We've been finding it every week, basically, since March, but sort of understanding what the utility of that is and how that can help inform the public health response. So for the next 10 minutes or so, I will talk about the trends in our data and try and understand how that compares to the COVID outbreak in King County. So this is just positive for SARS-CoV-2 over time using both the N1 and N2 targets. And this is just our skimmed milk method. So at the bottom here is Brightwater. That's had much more consistent detection for SARS-CoV-2 than the other two plants, which is a bit surprising given its smaller water catchment area. So if you remember from that map at the beginning, Brightwater had about 200,000 people compared to 700 and 800,000 people for West Point and South Plant. That part, I'll go back to this map too. So Brightwater collects samples from up here, and it also has a much lower burden of COVID. So there could be something here to do with how Brightwater, their treatment plant, is set up. And then South Plant has also had pretty consistent detection, which makes sense because they have a pretty high COVID burden in South Seattle and South King County. And then West Point has been all over the place. So that's a little bit harder to interpret, at least looking at it like this. So then the next thing we did was actually convert these CQ values into gene copy. So this is all of our methods. So the green line at the top is the limit of quantification, and then the dashed green line at the bottom is our limit of detection. So pretty much all of our data are above the limit of detection, which is good. But none of these values are above our limit of quantification. So we don't think that this means that it's not there. It just means that the error around those numbers is pretty wide. I tried to put error bars on this, and it was really messy and hard to see, so I decided to take them off. But so how we actually got the limit of detection, we ran 10-fold serial dilutions of the standard control, the IDT plasmid, in extracted Seattle wastewater, and then the limit of detection is 9 out of 10 replicates that are positive, while the limit of quantification is the lowest dilution with a coefficient of variation below 35%. So we took an average of all of the CQs and then calculated the coefficient of variation. So basically what this means is that numbers between these two lines have a pretty high degree of uncertainty in what that point value actually is. And the numbers below the limit of detection have an even higher uncertainty. And then, oh, too far. So this is just what those standard curves actually look like. As this points out, the gray is an error around the regression of all of the dilutions and the points. And as we get more dilute, we get a lot higher error in those values. And the blue line uses all of the dilutions and the red line uses only the first three. So our actual limit of quantification is a little bit, the number of concentrations we can use is much lower. So then when we graph these next to the positive clinical tests, and this is just for Seattle, and these values are every 2 weeks. Unless we get like permits for other data, they only give it as every 2 weeks. And this is also for all of Seattle, so it might not be representative of what is happening in each water catchment area. So we do see the really large spike in cases around the holidays, as well as the spike in cases right after Memorial Day here in June. We don't actually capture the large spike in cases in our wastewater data, but this could be due to much higher amounts of rain in Seattle during the winter months. So if we break the year into two sections starting on October 1st, the average daily flow rate is 40 million more gallons after October 1st compared to the first 9 months of the year. Since these plants already process, and this is data from West Point, I don't yet have data from South Point or Brightwater, but it's likely that they are experiencing something similar with the weather. So since the plants already process millions of gallons a day, any further dilution of that water could make it more difficult for us to detect SARS-CoV-2 at the treatment plant level. But there might be some more correlations in the summer. So I'll sort of zoom in on both of these time periods to try and look a little bit more closely. So this is just the N1 gene copy for the summer. So this looks like it could have preceded that spike in cases for South Plant. But like I mentioned before, there are some pretty big limitations right now to how this data are available. Again, this is not broken down by water catchment area. That's something that I hope to do soon. But it is possible that we do see a spike in South Plant before we start to see those clinical cases. For the winter, it might not be as clear. We do also here start to see a higher peak in cases compared in the N1 gene copy in our wastewater compared to those clinical cases. But like I said before, there's a lot of rain, so that could have affected our ability to find this. So we think that this might be a good reason to use flow rate as a way to sort of normalize this data to understand what the actual SARS-CoV-2 amount is in the water, and this would require us to collect 24-hour composite samples. One other thing I want to point out here as well is that all of these graphs use a linear scale for the gene copy, but all of the standard curves are made in log scale. So if we were to make that scale log scale, a lot of these trends here wouldn't look as peaked as they do here. And that's just sort of one thing to keep in mind that we've been thinking about as we look through all of our data as well. And then also with Seattle, as with most places, there's a pretty large disparity in testing, depending on the specific community that people live in and their socioeconomic status and the racial makeup of that community. So this map here is the rate of testing by health reporting area. So there's a much higher rate of testing in downtown Seattle and then in northern Seattle health reporting areas. These tend to be communities of higher socioeconomic status. They're more white than places in South Seattle. But places in South Seattle and then in rural King County have much lower rates of testing. However, when you compare that to this map here, which is the rate of positive cases by health reporting area, there's a much higher burden of SARS-CoV-2 in those South Seattle and South King County communities. So wastewater sampling could be particularly useful in these places where there is a lower rate of testing, but a much higher burden of SARS-CoV-2. However, we need to be thinking about sampling on the right scale. So this is just a transparent map like I showed before, showing those water catchment areas. So it's likely that sampling at treatment plants, because the scale is so large, might not be the best. So we're trying to look at getting into pump stations as well to get a better idea of community level SARS-CoV-2 burden. So the next steps that we're thinking of doing and are hoping to answer some more questions, we're thinking a lot about how we can normalize the data. And like I mentioned before, we're going to look at using flow rate as a potential way to look at the SARS-CoV-2 data in those treatment plants. We're also continuing with the recovery organism comparison to understand which one of those best models SARS-CoV-2 in water. A lot of groups are using bovine or mouse hepatitis virus. They're using bovine viruses, so we're just trying to understand how those each relate to recoveries. We're also doing an extraction comparison to understand which kit might be best in water. And then we want to do some more methods as well, because that first methods comparison did not capture all of the methods that people are actually running. And then we're also hoping to switch to digital PCR. Our department is in the works of buying digital PCR machines, so we'll be pivoting to using that for detection as well. And then all of this is done with the utility in mind of those, how public health response can actually use these data. So with that, I will finish up. I want to give a big thank you to everyone in the Environmental and Occupational Health Microbiology Lab. This has been a huge lift, and I'm very thankful that people have been here to help. This work was also partially funded by the Population Health Initiative and department funding. And then also we couldn't do this without the treatment plan sampling team. And then there's my email there if anyone wants to follow up on anything.
Thank you, Sarah. Great job. Very interesting talk. We have a couple questions in the Q&A, and I encourage others to add some questions. I have some too, in case you guys don't add any questions. Great job. So the first question we have is, have you been sharing any of your data with the wastewater treatment plants or county public health or the state, and how do you do that, and what has their reaction been?
Yeah, so we about every month meet with the wastewater treatment plants to present data. They're mostly just interested. We don't think that the SARS that we're finding in the treatment plants is viable. So on a risk level, we're not super worried about that. And then we're also working with the state health lab. They're trying to bring up a wastewater surveillance program right now. So we're helping them validate. We trained them on our methods. So they are interested because they're trying to start doing this as well on a whole state level.
Okay, thanks. I'm going to interject one of my questions here, which is, can you tell us a little bit about the sewage infrastructure in Seattle? Is it CSOs or SSOs? Are people all on sewage networks? Is there septic tanks?
So Seattle, yes, we do have combined sewer outflows. That's something that I've thought about of sampling after big rain events, is collecting there to see if there's any discharge of SARS-CoV-2 into the waterways. In Seattle proper, there's not a lot of septic tanks. Pretty much everyone is on the main sewage system. As you get into more rural King County, there are septic systems there. So obviously this doesn't apply for those more isolated communities. One other thing is we also have a pretty large homeless population. And I know that there's a problem with, they're allowed to park RVs in certain neighborhoods, and a lot of them just discharge their toilets into the rain drains. So that's also a concern as well, because that's not getting captured by the wastewater treatment system.
Thank you. The next question we have is, do you know of any publications to share about the disparity in COVID-19 testing? Any research that's been done to show where and why there are disparities?
So I don't have the name of publications off the top of my head. I know a lot of some more epi-focused research has been looking at this a lot. A lot of it has to do with access to healthcare facilities and just people not knowing where to go get tested. Some of it too is just availability of testing. It's really easy to get a test as a student at UW. That isn't always the case for people who don't have access to large university infrastructures. I'm happy to send some along if that person wants to email me. I can't think of the papers I've read off the top of my head, but I'm happy to share those.
Okay, that was Colleen Naughton at UC Merced, so you can connect with her.
Okay.
And then we have time for one more question, which was submitted, and it's around the issue of the LOQ and less than the LOQ, higher than the LOQ, and this is a problem that permeates all research focused on rare targets and environmental matrices, and all of us deal with this. Given your experience with this issue on this particular project, what do you think about measurements below the LOQ? Do you feel that there are certain conditions under which you can use them and certain conditions when you can't? Do you look at query your negative controls to help you decide that? What are the things that you've been thinking about with respect to this issue?
Yeah. So whenever we run PCR, we run all of our samples in duplicate. So usually if I get 2 hits on both of those show up positive at roughly the same place, that would make me more confident that it's actually there and that's more representative of what it is. We also run all of our samples with a 10 to the minus 1 dilution to see if that is representative as well. Unfortunately, a lot of the times we're working already at the tail end of what's there. So the 10 to the minus 1 dilution, that could indicate if it's inhibition or if we're at the limit of what's there. And the way we've been sort of approaching it is that there's just a lot of the range of what could be actually there for gene copies is really large. So we haven't really been presenting gene copy data to the state public health labs in the treatment plants, just because we're not confident to be able to say that, oh yeah, there's 600 copies of the virus there, because we don't know, because we're in between those two values. But a lot of it just has to do, and then because we've been running multiple methods each week on the sewage, it's likely that we get multiple detections across methods. So that would also make me more confident that what's there is actually there.
Thank you for sharing some thoughts on that. And like I said, this is a question that is not only important to the environmental virology community, but everyone working on pathogens in the environment, people working on eDNA for detection of invasive species. So it is a really important issue. So it's a challenge that we have to face. Thank you very much, Sarah. Great job with your presentation and answering questions. You can go ahead and stop sharing your screen, and I'll ask Bo to start sharing his screen. As I mentioned before, Bo is a postdoc at University of Hawaii, and it is 2 hours earlier there, so it's 6:30 in the morning. So thank you for joining us so early, and Jill is going to be the moderator for Bo. You can put it on, oh, you got it, presentation mode. Great.
Good morning, everyone. Thanks for your attending this morning. My name is Bo. I'm currently a postdoc in the Civil and Environment Engineering in University of Hawaii at Manoa in Dr. Yan's lab. We really appreciate this chance to present our recent lab work about the fine-scale temporal dynamics of SARS-CoV-2 RNA abundance in wastewater during a COVID-19 lockdown. This research is done in the city and county of Honolulu. The city county of Honolulu is the consolidated city and county in the state of Hawaii. It includes both the city and the city of Honolulu, which is the state capital and also the largest city. And it also includes the rest of the island of Oahu. And the entire county with a population around 1 million. Since the first reported case in April, Oahu is always the outbreak of COVID-19 in the Hawaii. Around 80% of the total statewide cases were reported in Oahu. In last August, we have experienced a rapid surge in the clinic case with a 7-day average more than 200. To control the outbreak, the government has issued a 2-week stay-at-home order starting from August 27th. And this order was continued by another one for another 2 weeks. So we have experienced a continuous 4-week lockdown in Honolulu. As the result, we were expecting a rapid decrease in the COVID-19 clinic case. And this gave us a very rare opportunity to see how the wastewater SARS-CoV-2 concentrations respond to the decrease in clinic case. In total, there are 9 different wastewater treatment plants in Honolulu. But majority of the wastewater from the entire island are treated by the 2 largest ones. The Sand Island treats more than 55%, and the second largest one, Honolulu, treats 27%. And the surface area of the 2 wastewater treatment plants covers all those populated communities in Honolulu. So we mainly focus on wastewater from the influence of these 2 largest wastewater treatment plants, the Sand Island and Honolulu. Starting from the first day of lockdown, we have continually sampled daily flow adjusted wastewater for 39 days. This sampling period covers the entire 4-week lockdown and also with 10 days extension. As we were interested in if the liquid and solid fraction of the seawater will show the similar trend or dynamics. So at first, we separated our wastewater sample into solid and liquid fraction by centrifuge. And then the liquid fraction was treated by a PEG method and to collect the pellet. Then the solid fraction and the pellet from the liquid were extracted RNA. The RNA were synthesized into cDNA with using superscript for reverse transcription kit using random Hapmer. Then those cDNA were used as DNA template for our targeting with interest by real-time qPCR. We have tested 3 different assays for SARS-CoV-2, including the N1, N2 gene recommended by the CDC and also the E gene recommended by the WHO. To account for the different fecal strands in wastewater and also taking consideration in about the micro steps variation, like centrifuge or reverse transcription, we have also measured 3 endogenous fecal RNA viruses, include the F-specific RNA codiphage group 2, group 3, and also a compromised motor virus, the PMMOV. The 3 RNA viruses are commonly used as surrogate pathogens to see their persistence in wastewater. And we also used the 3 different thick iron viruses as global limitation of our SARS-CoV-2 concentration. And before we test our wastewater samples, we have tested method reproducibility by treated 6 samples, both liquid and solid fraction, processed in triplicate, and then we compare the detected CQ value by qPCR from those triplicate process. The variation of those detected CQ from the triplicate process shows a very limited variation, and the delta CQ in liquid is only around 0.5 cycles, and this number is about 0.6 cycles in the solid fraction. So we think the overall method reproducibility is very reliable. But then we further conferred the assay specificity by a sequencing of some samples. We have run triplicate qPCR for several samples and mix the triplicate qPCR product from the cell assay into single tube, and then we display the mixed product on gel electrophoresis. From this gel figure, we can see for each target gene, we can see very clear band at their cracked DNA size, and then we further sequence those mixed triplicate PCR product by next generation sequencing. Use Illumina millisec platform. So for the sequencing result, we have first deployed those sequence by data to remove chimeric structures, and then we align those treated secondaries aligned to our target by using BLAST. From the bioinformatics analysis, we found target sequence were detected in all those qPCR products, and the similarity to their respective target gene is with coverage higher than 100%, and with identity also around 100%. So we can say the assay specificity applied to wastewater sample are quite reliable. As a result of lockdown, since the start of lockdown, we have seen a rapid decrease in the clinic case, both the daily case and the 7-day moving average. The 7-day moving average decreased from the beginning of more than 200 to 89 at the end of lockdown, and this number goes lower to only 41 at the end of our sampling campaign. So we have looked into the 3 endogenous virus, RNA virus in our wastewater samples. Generally, we didn't see any obvious trend, no increased trend and no decreasing trend in the concentration of the fake RNA viruses. And all those RNA viruses concentrations showed at a relatively stable concentration level, although there are some significant variation in some daily samples. And we have also compared those concentration levels to the public data. Our concentration levels are comparable to those reported values. And we also compare the results from the 2 wastewater treatment. And the concentration levels from Sand Island and Honolulu are pretty similar. This slide is about the overall dynamic of wastewater SARS-CoV-2 in our wastewater sample. The left panel is from the Sand Island, and the red panel is from the Honolulu. Different from the fake indicators, we have seen the wastewater SARS-CoV-2 showed correspondence with the decreasing clean data. We can see very clear decreasing trend in both the solid fraction as shown in the blue line, and also the same decreasing trend in the solid fraction as shown in the red line. And this decreasing trend can be seen in all those three different assays: N1 assay, N2 assay, and the E assay. In the wastewater treatment plant, as shown in the right panel here, we can also see a similar trend, decreasing trend in solid fraction, the same decreasing trend in the liquid fraction, and the N1 assay, N2 assay, E assay all show the decreasing trend. Besides the overall decreasing trend in those three assays, we have also observed a significant daily fluctuation in those wastewater SARS-CoV-2 concentration. Even though in the same week, take the ESA from Sand Island as example. On day 4, the difference between the day 4 and day 6, they are samples from the same week, but difference is larger than 2 log numbers. So we think if we want to track the temporal dynamic of wastewater SARS-CoV-2 concentration, considering its high significant daily fluctuation, we need to take the fine scale sampling frequency, such as daily sampling frequency, to track this dynamic. Here we also compare the performance between the different assays. The results from the liquid samples are shown in the left panel. Overall, among those different assays, they showed a fairly good correlation between each other, and all with R value 0.7, 0.7, and 0.8. We found the best correlation is found between the N2 assay and the E assay. In the solid fraction, we also observed the similar observation. We found a fairly good correlation between different QPCR assays, and the best correlation was also found between the N2 and the E assay. Here we have compared the correlation between the SARS-CoV-2 in solid and in the liquid. Here, in this figure, we put all those data from the two wastewater treatment plants together, as they show no significant difference. Here, from this figure, we can see overall by all those three assays, we can see relatively good correlation between the solid concentration and the liquid concentration. With all R value, N1 is 0.6, N2 is 0.8, and E R value is 0.7. And compared to N2 and the ESA, N1 shows a less consistency in the solid and liquid fraction. And then we calculate the mass distribution in the solid and liquid fraction by using the detected concentration and also the solid content from each wastewater sample. By this result, we have observed a similar mass distribution as reported by many previous studies before. We found that the majority of the virus RNA present in the solid part. By all those three different assays, N1, N2, and E, we found the mass percentage in solid is more than 80%. Here we, to account for the variation in fecal strength and also variation in the multi-step process of our samples, we have normalized our SARS-CoV-2 abundance to the three different fecal indicators, normalized by G2, normalized by G3, normalized by PMMOV, and after the normalization, we still found the decreasing trend in all those three assays still remained. Such as N1 assay, we can see a significant decreasing trend in correspondence with communicase, and we found N2 assay in both liquid and solid, as shown in the blue line, the red lines, we see the clear decreasing trend. And in E assay, we have a similar observation. And by and in the normalization by different figure indicators, we have the similar decreasing trend, such as normalized by G2 in ESA. It shows very good linearship about the decreasing trend, and normalized by G3, here it's also significant decreasing, and by PMOV, the decreasing is also significant. So here, from all those results we have above, we can draw a conclusion that the downward trend of SARS-CoV-2 RNA abundance, both the measured concentration and the normalized abundance to different figure indicators that are corresponding to the rapid decrease of clinical cases during the lockdown in Honolulu. And the second one, we have found the SARS-CoV concentrations in liquid and solid fraction were correlated to each other, and they showed a similar trend and also similar daily dynamics. The other point we found is that we have observed significant daily fluctuation of the wastewater SARS-CoV-2 RNA abundance. Even with the same weeks, the difference between two days maybe larger than 2 log numbers. So this suggesting the file scale sampling frequency, such as daily sampling, is needed to take into consideration when we try to track the dynamic of SARS-CoV-2. Besides those results we have obtained, we are also interested in answering some questions, such as to understand what the driving factor behind the daily fluctuation. If it's because of the different decay rate in the pipe system, or it's because of the different virus shedding in the fecal from patients during different infection periods. And then we also interested in to study correlation with daily clinical case numbers in the 2 severe sheds of the 2 wastewater treatment plants. Currently, the clinical data we have is from the whole city, but actually the Sand Island, Honolulu, they have their specific service area. So we are trying to obtain that clinical data from their specific service share. Then we can do cross correlation to between the clinical case and their respective SARS-CoV-2 concentration in the wastewater treatment plant. The project is mainly supported by a grant from the National Science Foundation, and it's also partially supported by the city and county of Honolulu. Most of the lab work and data analysis was done by with the help of our group members, including Dr. Dean, Prakit, Minky, and Dr. Yan. And we need to thank Lyle and Gayatri and Sharon for their assistance with sample collection. And we also need to show thanks to Joshua for his assistance with obtaining the clinic data. That's all. Thank you for listening. And any question is welcome.
Thanks, Bo. That was a great presentation. Obviously, a lot of really hard work went into that. So thank you so much for sharing. A reminder, if anybody else has questions, please add them to the Q&A box, and I'll be starting to ask the ones that are already there. All right, so our first question here is related to how you're sharing your wastewater data. Are you sharing that information with the treatment plants, health departments, or the public in Hawaii?
And actually, we have built a long-term wastewater surveillance about the SARS-CoV-2, and all those data are available at the website, oneor.org dashboard. And it's not only the data from the Sand Island and Honolulu. Actually, it's all the data from the 10 wastewater treatment from the entire Honolulu County.
Great, thanks so much. That'll be really interesting to take a look at too. All right, our next question is about your normalization targets. So you mentioned, that by normalizing your concentrations, you were still able to see that decrease in concentration overall. We have a question wondering if those targets are, if the variability is a true variation in wastewater strength, or if it's just a variation in the targets themselves. Basically, do you have a sense for that? And did you use any other metrics for wastewater strength, like flow rate or TSS as well?
Yeah, we have also monitored the wastewater flow. Here is the wastewater flow from the 2 wastewater treatment plants during our sampling period every day, the daily water flow. And actually they are kept at very stable levels, exactly same with a variation less than 5%. And the Honolulu, it uses a separate system to collect rainwater and wastewater. So we believe the concentration in our result is a real concentration other than the effect by rainwater or fluctuation of wastewater flow. About the TSS, yes, we do measure the TSS in each sample, and the data is not shown here, but we have found that TSS is very stable in the 2 wastewater treatment plants.
Great, thanks. And I have a sort of a follow-on question related to that. So I might have missed it in your presentation, but you mentioned, basically you showed the original data for the SARS-CoV-2 markers, and then also the normalized data. And both of those data sets showed a decrease in concentration over time, right?
Yeah.
So basically I'm wondering if you think normalization is necessary. If you were able to see the trends without normalizing, was there an improvement in the fit or a reduction in variability when you normalized, or were the trends that you observed across those 2 data sets, unnormalized and normalized, fairly similar?
Yeah, from the result I presented today, with the convolutional control is that after with normalization or without normalization, we can see the same decreasing trend. And there is another work we are doing now. We have did cross correlation about the clinical data and with the normalized SARS-CoV-2 concentration and those unnormalized SARS-CoV-2 concentration. We do have found some correlation between the clinical data and the wastewater SARS-CoV-2, and this correlation is significant. And we have found among the 3 different normalization by G2, G3, or PMO, we found by normalized to G2, we can improve the relationship of cross-correlation.
Great, thank you. Let's see, are there if anybody else has more questions, please add them to the Q&A. I'm just looking over my notes to see if I had any other specific ones here. But I don't think so. So, oh, here's one. So some of this was obviously a lot of work to do this daily sampling during, about a month-long period, I think it was overall, or more. And you mentioned that you're going to look into what's affecting the daily sampling variability, right? From day to day, you can see pretty changing concentrations. I'm curious if you think after doing this work, what your thought would be, what your recommendation would be on an ideal sampling frequency that's maintainable across a long time. Do you think that we really need to be doing this sampling daily because the fluctuations are so high, or do you think that a reduction in sampling frequency is still feasible to get good data, since a lot of people, workload and processing time might be tricky?
Yeah, yeah, that's a very good question. Yeah, then besides the daily fluctuation, and we have also compared the 7-day moving average of the SARS-CoV-2 concentration and the 7-day moving average of the clinical data. We have from our result, we can see the similar trend. So, so that means even though by like weekly sampling, we can still track the overall trend. But if we want to see the temporal dynamics of communicate, like what's the difference between day and day, then we need to consider daily sampling frequency.
Great. Yeah, it's a tough balance, I know, and we're all still sort of figuring that out. Well, great. If there are no other questions, I think we can close. I did want to make one quick announcement before we finish. I mentioned in a previous webinar that we are looking for students who are interested in submitting training videos on methods, data analysis, grad students or undergraduates. I'm going to paste the link to the details for this in the chat. And if you would share this with any students that you know that are interested, we're looking for just a short mini proposal on what students would like to do. That would be due next week, March 10th. So I just wanted to give one more quick plug for that. And otherwise, I want to thank our presenters, Bo and Sarah. And we'll have another webinar coming up in another couple of weeks. So thank you all so much for joining us, and have a great rest of your days.